Hello dear students, myself Bhavani Pentela, working as an assistant professor in Noida Institute of Engineering and Technology, Pharmacy Institute. In this lecture, we are going to study about neurohumoral transmission in C.N.S. and we will study about some important neurotransmitter like GABA and Glycine. So, without any further delay, we will dive into the session. So, we will start here what is meant by neurohumoral transmission in C.N.S. Before going to the neurohumoral transmission in C.N.S. steps, let me see about what is a neurohumoral transmission. So, what is a neurohumoral transmission? If you see here the term neurohumoral transmission. So, the transmission, the name itself indicates something is going to transfer, right. So, what is going to transfer here? The nerve impulse from the presynaptic neuron to the postsynaptic neuron with the means of humoral agent. Okay. With the means of humoral agent, then nerve impulse is going to transfer from the presynaptic neuron to the postsynaptic neuron, then it is called as a neurohumoral transmission. Okay. So, what is the neurohumoral transmission here? So, neurohumoral transmitters, so what about the neurotransmitters? So, whatever the neurotransmitters like excitatory neurotransmitter or inhibitory neurotransmitter, whatever it may be, first it will store in the storage granules. When it receives the impulse, now the neurotransmitters from the storage granules, it will be released into the synaptic cleft. After releasing into the synaptic cleft, it will go and bind to receptors which are present on the postsynaptic neuron and it will elicit the action. This is the main phenomena that what the neurotransmitter will do whenever it releases from the storage granules. Okay. So, what is the life cycle of neurotransmitter? In this slide, we will learn what is the life cycle of neurotransmitter. So, we had known about the neurotransmitters. The neurotransmitters are of two types category which you can name as excitatory neurotransmitter and inhibitory neurotransmitters. They are the both neurotransmitters like which having both excitatory in action and inhibitory in action. Okay. So, here we will see about the life cycle of any neurotransmitters. So, what is the life cycle? What it is going to do here? First one, it have to synthesize. First, it have to synthesize in our body. So, whatever the neurotransmitter like acetylcholine, adrenaline, non-adrenaline or dopamine, serotonin, whatever it may be, GABA, glutamate, whatever it may be, first it have to synthesize in the body. So, first step was synthesis. Okay. After synthesis, it will store in the storage granules. After synthesis of neurotransmitter in the body, it will store in the storage granules. Okay. Second step was storage, right. And after the storage, when the impulse has been generated, here I am saying when the impulse has been generated, now the synaptic vesicles, so now the neurotransmitters which are stored in the storage granules, now it will be released into the synaptic cleft. After generation of nerve impulse, first what is going to happen? First the neurotransmitter is going to synthesize and second one it will be stored in the storage granules and once it receives the impulse and it will be released into the synaptic cleft. After releasing into the synaptic cleft, now this neurotransmitter will go and bind to the postsynaptic neuron. Now, where it will go and bind here? It will go and bind to the postsynaptic neuron, where the neurotransmitter receptors has been present. For example, if the neurotransmitter is acetylcholine, so what the receptors will be present on the postsynaptic neuron? So, the receptors will be the muscarinic or nicotinic. For example, if the neurotransmitter is like adrenaline, so what the receptors will be present on the postsynaptic neuron? Alpha and beta. Depending on the neurotransmitter, what is going to release on the synaptic vesicles is the specific receptors will be present in our body and it will go and bind to its specific receptors which are present on the postsynaptic neuron. At the same way here, when it released into the synaptic cleft, what is the fourth step is going here? The fourth step was it will go and bind to the postsynaptic receptors. So, postsynaptic receptors, when it bind to the postsynaptic receptors, now these receptors will elicit the action. Now, what will going to happen? The receptor and neurotransmitter complexation will be formed, right. The drug and receptor complexation will be formed and this complexion will elicit the response. And finally, when it shown it response, it have to terminate from there. Right, you have to terminate from there. How it will be terminate from there? It will be terminate by the metabolism of some other enzymes or it will be again recycled into the presynaptic neuron for the further synthesis. So, this is the basic 5 steps or the 5 life cycles which you can see with the neurotransmitter. What is the first thing? First thing was synthesis of a neurotransmitter and second thing was storage. After synthesis, it will be stored in the storage vesicles. After staying in the storage vesicles, once the neurotransmitter has received the impulse, then it will be released into the synaptic cleft. After releasing into the synaptic cleft, these neurotransmitters neurotransmitter will go and bind to the postsynaptic neuron where the receptors has been present. Once it go and bind to the postsynaptic neuron, when it shows its action, it will be terminated from there, right? It will be get terminated from there, and finally the steps will be happen. What is the action will be shown with the neurotransmitter. And this is the small diagrammatic way to understand in a better way. So, if you see here, so this is an presynaptic neuron, right? This is an upside portion, this is an presynaptic neuron. The space between here, this is called as an synaptic cleft. Okay. So, what is a synaptic cleft here? I think you can see in a better way here, this is a presynaptic neuron and this is a postsynaptic neuron. The space between the presynaptic neuron and the postsynaptic neuron is called synaptic cleft. Okay. Once the receptor has generated the impulse, okay, before generation of an impulse, first just in the previous slide I have explained you, here the neurotransmitters will be stored in the storage granules. So, first the neurotransmitter will get synthesized in your body and it will be stored in the storage granules. So, what is this one? This one is the storage granules. Once the storage granules, once the receptor stored in the storage granules and when it receives the impulse, now the storage granules inside the neurotransmitter will released into the synaptic cleft. So, the space here, this is called as a synaptic cleft. Once it released into the synaptic cleft, it go and bind to its receptors. Right, it will go and bind to its receptor and it will shows its action, post junctional action, and finally the neurotransmitter will be terminated from the site. Okay. So, for example, if I am saying this one is an acetylcholine neurotransmitter, okay, this is an acetylcholine neurotransmitter. So, when it generated the impulse, when the impulse has been generated, now this acetylcholine from the storage granules, it will be released into the synaptic cleft. And once it released into the synaptic cleft, it will go and bind to its receptors. Which type of receptors? Two types of receptors for the acetylcholine. What are they? One is muscarinic and second one is nicotinic. So, it will go and bind to the muscarinic receptor and nicotinic receptor to show its action. Once the post-junctional action it has been shown, it will be terminated from there. If I am talking about this muscarinic receptor and nicotinic receptor, both are the receptors in a different category. One is an G protein coupled receptor and second one is an ion channel receptor respectively. Muscarinic is an GPCR receptor and nicotinic is an ion channel receptor. Depending upon the receptor which the acetylcholine go and binds, it will shows its action. Okay, this is the first example. So, if I am saying this is an acetylcholine, it is a neurotransmitter. If I am talking about glutamate, okay, if I am talking about glutamate neurotransmitter, glutamate is also a neurotransmitter. It is a powerful excitatory neurotransmitter. Which type of neurotransmitter is glutamate? It is a powerful excitatory neurotransmitter. The same thing will happen here. It will be stored in the storage granules. Once it receives the impulse, now this glutamate will be released into the synaptic cleft. And when it released into the synaptic cleft, this is the first example which I had explained you, and this is the second example, this is called glutamate. Okay. So, glutamate. So, when the glutamate has been released into the synaptic cleft, now it will go and bind to the receptors for the glutamate neurotransmitter. So, what are the receptors for glutamate? There are the two types of receptor. What are the receptors there? NMDA receptor and AMPA receptors. Okay. So, there are two types of receptors for glutamate on the post-junctional neuron. So, when the glutamate will be released into the synaptic cleft, it will go and bind to the post-junctional neuron, and their receptors are present on the post-junction. So, what the receptors are present for the glutamate neurotransmitter? NMDA receptor and AMPA receptor. So, it will go and bind to this particular receptor to show its action. Once it show its action, it will be terminated from there. So, this is the second example. You can take the third example as well. For example, if I am taking about the GABA. So, GABA is also in a neurotransmitter. Which type of neurotransmitter it is? It is an inhibitory neurotransmitter, right? It is an inhibitory neurotransmitter. The same thing will be happen here. Once it releases from the presynaptic neuron from the storage granules, it will released into the synaptic cleft. Once it released into the synaptic cleft, it will go and bind to its receptor. What type of receptors for the GABA? GABA we having three types of receptors, namely GABA A receptor, okay, and GABA B receptor and GABA C receptor. Depending upon the receptor which it will shows its action, according to that the neurotransmitter will go and bind to its particular receptor to show its action on the postsynaptic neuron, and finally the neurotransmitter will be terminated from there. So, this is the few example which I can explain you in a better way, right. So, now we will see about the next one, excitatory and inhibitory postsynaptic potential. So, what is an excitatory postsynaptic potential and what is an inhibitory postsynaptic potential? So, look at here EPSP and IPSP. So, excitatory postsynaptic potential which you can term as EPSP. EPSP is a short name. For which it is a short name? It is an excitatory postsynaptic potential. And what about IPSP? IPSP is represents inhibitory postsynaptic potential. So, I will explain one by one here. First, I am moving to the EPSP receptor. So, what is an EPSP? EPSP is an excitatory postsynaptic potential. Okay. Now, once the glutamate excitatory, so the name itself indicating excitatory, here the excitatory neurotransmitter will go and bind to the postsynaptic neuron and finally it will leads to the depolarization. How it will leads to the depolarization? Once the glutamate neurotransmitter go and bind to its postsynaptic neuron, then the opening of sodium and calcium channel will be happen. By the opening of this sodium and calcium channel, ultimately it will leads to the depolarization. I will explain you one more time. EPSP. EPSP. What is an EPSP? EPSP is an excitatory postsynaptic potential. What is an excitatory postsynaptic? So, excitatory neuron is going to show its action on the postsynaptic neuron, right? So, what is an excitatory neurotransmitter here? Glutamate. Glutamate is an excitatory neurotransmitter. Glutamate is an excitatory neurotransmitter. So, once it released in the synaptic cleft, so this is an synaptic cleft. So, once the glutamate neurotransmitter, once the glutamate excitated neurotransmitter released into the synaptic cleft, it will go and bind to the postsynaptic neuron. Once it go and bind to the postsynaptic neuron, I had said in a previous slide, for glutamate we having two types of receptors which are named as NMDA receptor and AMPA receptor. NMDA receptor is responsible for opening of calcium channel and AMPA receptor is responsible for opening of sodium channel. Now, when the glutamate go and bind to its particular receptor or specific receptor, what is going to happen? Opening of sodium and calcium ions or sodium and calcium channel. Now, once the glutamate go and bind to its receptor, so what channel is going to open here? The both channel, sodium channel and calcium channel. Sodium channel, when the sodium channel will get open? The sodium channel will get open when it will bind to the AMPA receptor. When the calcium channel will get open? When it will bind to the NMDA receptor. So, this is the 2 points, it will be clear, right. So, glutamate having two types of receptor, namely AMPA receptor and NMDA receptor. When the glutamate go and bind to the NMDA receptor, the opening of calcium channel is seen, and when the glutamate go and bind to the AMPA receptor, the opening of sodium channel is seen. And finally, what is going to happen here? Depolarization of an cell. So, this is called as an excitatory potential, excitatory postsynaptic potential. Now, I am talking about the inhibitory postsynaptic potential. What is about an inhibitory postsynaptic potential? So, here inhibitory neurotransmitter. What is an inhibitory neurotransmitter here? I am taking an example, GABA. Even glycine is also an inhibitory neurotransmitter. Okay. So, if I am taking this neurotransmitter GABA, gamma aminobutyric acid, it is an inhibitory neurotransmitter. When the inhibitory neurotransmitter released into the synaptic cleft and go and bind to its postsynaptic neuron, then what action it is going to show here? It causes the hyperpolarization. How it causing the hyperpolarization? It causing the hyperpolarization by opening of chloride channel. Here you have to notice the 2 points. The excitatory neurotransmitter when and go and bind it to its receptor, opening of sodium channels and calcium channel has been seen, which will leads to or which will result in depolarization. When the inhibitory neurotransmitters go and bind to its receptor, so then what is the action is going to seen here? Hyperpolarization due to because of opening of chloride channels. Okay. The glutamate is responsible for opening of sodium and calcium channel, GABA is responsible for opening of chloride channel. So, due to because of glutamate you can see the depolarization, and with the GABA you can see the hyperpolarization. So, next we will move to the chemical mediators in CNS. So, there are the 4 chemical mediators in CNS, namely as biogenic amines, amino acids, neuromodulators and lipid mediators. So, there are the 4 mediators which are seen in the CNS. What are they? Biogenic amines, amino acid, neuromodulators and lipid mediators. These are the 4 chemical mediators which you can see in the CNS. If I am talking about the biogenic amines, what are the biogenic amines? Biogenic amines are the serotonin, histamine, adrenaline, non-adrenaline, dopamine, all these falls under the biogenic amines. So, what are the amino acids? Aspartate, glutamate, GABA, glycine, all these falls under amino acids. And what about the neuromediators, neuromodulators? Neuromodulators are neuropeptides, substance P, these are falls under neuromodulators. What about lipid mediators? Lipid mediators, the lipid, it will be generated from the lipids, lipid bilayer. So, what is the example falls under the lipid mediators? Prostaglandin. Okay. So, if I am talking about these two one, serotonin and histamine, both are the indolamines, serotonin and histamine and the indolamines, adrenaline and non-adrenaline are the catecholamines. So, these are the chemical mediators in the CNS. Okay. So, here we having the 4 steps in the neurotransmitters. So, what is the basic process in the neurohumoral transmission in CNS, right? This is the main topic in our syllabus, especially in the 4th unit. Okay. So, there are the 4 processes, there will be the 4 processes occurs with the neurotransmitters. What are the 4 processes you can see here? First one is neurotransmitter, second one is neuromodulator, third one is neuromediator and fourth one is neurotropic factors. So, these are the 4 factors. What are the 4 factors? Neurotransmitters, 4 process, second one is neuromodulator, third one is neuromediator and fourth one is neurotropic factors. Now, we will explain, I will see one by one in a better way. Okay. So, neurotransmitter. So, what is a neurotransmitter? Neurotransmitter, when it has been stimulated and it will go and bind to its receptor, it will shows its action on the postsynaptic neuron. Either it can stimulate the postsynaptic neuron or it can be inhibit the postsynaptic neuron. This will be seen with the neurotransmitter. Okay. So, if it is an excitated neurotransmitter, it will excite the cell. If it is an inhibited neurotransmitter, it will inhibit the cell. So, what is the example falls under the neurotransmitter? GABA and glycine. So, one is an excitatory neurotransmitter and second one is an inhibitory neurotransmitter. Second one is neuromodulators. So, what are neuromodulators? Neuromodulators are released by the neurons and it will produces the slower action on a pre and postsynaptic neuron. So, where the neuromodulators will be generated? Neuromodulators will be produced or it will be released by the neuron and it will shows the slow action on the presynaptic neuron or the postsynaptic neuron when it will be seen. So, what are the examples under the neuromodulators? Carbon dioxide, purines, peptides, okay, substance P, all these are the neuromodulators. Okay. Next one, neuromediators. So, what are the neuromediators? Neuromediators are the substances which will be released with the neurotransmitters and it will elicit its response. For example, if you see here cyclic AMP, cyclic GMP, inositol triphosphate. So, these are the neuromediators. Okay, neuromediators. Neuromediators are also called as an secondary messengers. Right? Neuromediator is also called as an secondary messengers which will plays a very crucial role. It will be shows its action on the postsynaptic neuron when it released by the neurotransmitter. So, neuromediators are also called as secondary messengers. What are the secondary messengers? Cyclic AMP, IP3, these are the secondary messengers. And what is the fourth process in the neurohumoral transmission on CNS? Fourth one is neurotropic factors. The major difference between the neurotropic and a neuromodulator was neuromodulator will be released by the neuron, it will shows the slower action, but in the neurotropic factors it will be released by the neuron, but the action will be the somewhat longer than that of any neuromodulators. Okay. So, what are the example falls under neurotropic factors? Cytokinins, growth factors, these are the examples which falls under neurotropic factors. So, these are the 4 factors. What are the 4 processes here? Neurotransmitters, neuromediators, neuromodulators and neurotropic factors. These are the 4 processes which you can see in the CNS. Okay. So, now we will see the some important neurotransmitters like GABA and glycine, both are the inhibitory neurotransmitter. So, if I am talking about the GABA neurotransmitter, GABA is an inhibitory neurotransmitter. It is an neurotransmitter which is present in CNS, which will shows an inhibitory in action. It will plays a very important role in behavior, conjugation and some stress emotions like some it will respond to the stress like emotions, learning, sleep. Okay. So, if I am talking about the receptors, the GABA GABA having 3 types of receptors. What it named as GABA A receptor, GABA B receptor and GABA C receptor. Okay, the GABA A receptor is an ion channel receptor, GABA B is an G protein coupled receptor, and finally GABA C is also an ion channel receptor. And if I am talking about these GABA receptors, these are composed, most of the GABA receptors are composed with alpha, beta and gamma subunits. Okay. So, this is the synthesis, how the GABA will be synthesized. With the starting material for GABA synthesis was glutamine. Okay. The glutamine, in the presence of glutamate synthesis, what is going to produce here? Glutamate. In the presence of glutamate decarboxylase, you will synthesize the GABA. So, this is the synthesis of GABA. What is the starting material for the synthesis of GABA? Glutamine. From the glutamine, you will produce the glutamate, and with the help of glutamate decarboxylation, finally you will get the GABA gamma aminobutyric acid, right. And next one, GABA neurotransmitter. If I am talking here, I will explain you what are the agonist drugs and what are the antagonist drugs for the GABA neurotransmitter. Okay. So, if I am talking about the GABA neurotransmitter, so most of the drugs like anti-epileptic drugs, sedative drugs, hypnotic drugs, anesthetic drugs, these are the main drugs, these are the drugs having the main target and will bind to the GABA receptor and it will shows its action, right. So, if I am talking about the drugs here, GABA receptor agonist drug was muscimol, and what is the antagonist drug for GABA A receptor? Picrotoxin, right. And if I am moving to the GABA B receptor, what is the agonist drug for GABA B receptor? Baclofen. And what is the antagonist drug for GABA B receptor? Saclofen. So, these are the drugs of GABA, some are the agonist drugs and some are the antagonist drugs to the particular receptor. Okay. Now, we will move to the second type of neurotransmitter, which is also an inhibitory neurotransmitter. It is also an amino acid neurotransmitter which is present mainly on medulla and spinal cord. So, it also shows an inhibitory action. Okay. It also will shows inhibitory action, especially it will prevent the desensitization of NMDA receptor. Okay. So, the function, what is the main function of glycine? Hyperpolarization. So, what is the main function of glycine? It is an inhibitor neurotransmitter, it will open the chloride channel. So, the function of the glycine neurotransmitter was hyperpolarization. So, here there are the some drugs for glycine. Agonist drugs are beta alanine, antagonist drugs are strychnine. Okay. So, next one, if you are moving to the glycine neurotransmitter. Glycine neurotransmitter we having, if I am talking about the glycine neurotransmitter, there will be the transporter for the glycine. So, there are 2 types of transporters for the glycine. One is named as glycine T1 transporter and second one is named as glycine T2 transporter. So, what is the glycine T1 transporter? Glycine T1 transporter is any primarily glial glycine. So, it is any primarily glial glycine. And what about the glycine T2 transporter? It is primarily glial neuronal. One is glial glycine transporter and second one is glial neuronal transmitter. So, this is about the importance of any GABA glycine neurotransmitter. Especially this glycine neurotransmitter will prevent the action of glutamate neurotransmitter. It will desensitize the receptors like EAMPA and NMDA. So, because of this receptor, there will be opening of sodium channel and calcium channel, which will causes the cell depolarization. So, what the glycine molecule, what the glycine neurotransmitter will do here? The glycine molecule, the glycine neurotransmitter have to show the inhibitory action. How it will shows the inhibitory action? It will shows the inhibitory action by opening of any chloride channel. How by opening of this chloride channel, it will we can see the hyperpolarization. In such a way, the glycine will shows its action. So, these are the 2 important neurotransmitters which falls under inhibitory neurotransmitters. Both are the amino acid neurotransmitters. If I am talking about the glycine, if I am talking about the GABA, both are the inhibitory neurotransmitter which falls under amino acid neurotransmitters. Thank you.